Thank you very much, Shinjini, for your kind introduction. And I also appreciate organizers for inviting me to such a nice symposium. I really enjoy staying in this city, and I am also happy to see my great teacher and our big collaborator of mine, Professor G.B. Nair, here in Bangalore. So today, my topic would be the probiotics, innate immunity, and intestinal inflammation. So, functional characterization of toll-like receptors have established that toll-like receptor-dependent recognition of microbes induces activation of innate immunity and subsequently induces activation of the development of the antigen-specific adaptive immunity, especially Th1 responses. So, this sequential activation of the innate immune system is actually required for the host defense against pathogens. So, our laboratory is now interested in the in vivo involvement of this innate immunity in several immune responses. So actually, innate immunity is essential for the infectious diseases. We are also interested in the involvement of the innate immunity in several immune disorders, particularly inflammatory bowel diseases. So, inflammatory bowel diseases in humans are represented by the Crohn's diseases and ulcerative colitis. And both diseases are the intractable diseases with unknown etiologies. And more importantly, particularly in our country, Japan, the number of patients with the Crohn's diseases and ulcerative colitis are both tremendously increasing in these 20 years. I'm not sure in the situation in India, but the same tendency can be observed in several Asian countries, such as Korea and Taiwan. So, in order to know the etiology of these inflammatory bowel diseases, variety of mouse models of the inflammatory bowel diseases have been so far developed. So actually this table was from the review in 2002, and that time the pathogenesis of the inflammatory bowel diseases were mainly discussed based on the aberrant Th1 or Th2, so-called adaptive immune responses. We have also developed the mouse models of the inflammatory bowel diseases using the mice lacking STAT3 selectively in innate immune cell populations. Since STAT3 knockout mice are embryonic lethals, we employed conditional gene targeting technologies to generate these mice lacking STAT3 selectively in innate immune responses. Through the analysis of these mice, in the absence of the IL-10 STAT3 pathways, actually STAT3 is essential for the IL-10 signaling in the innate immune cell populations. And in the absence of the IL-10 STAT3 pathways in innate immune myeloid cells, such as macrophages and dendritic cells, these innate immune myeloid cells are abnormally and continuously activated. And under these conditions, these innate immune myeloid cells produce high amounts of the inflammatory cytokines. Among these inflammatory cytokines, IL-12P40 is responsible for the aberrant Th1 or Th17 responses that causes the development of inflammatory bowel diseases. And additionally, in the absence of IL-10 STAT3 pathways, toll-like receptors can trigger aberrant production of these inflammatory cytokines through the recognition of the microflora in the intestine. That's why, actually, in the absence of STAT3, these innate immune myeloid cells were abnormally activated systemically, but inflammation can be observed only in the intestine. This is because we have microflora in the intestine that is recognized by toll-like receptors. So since then, we became interested in the innate immune myeloid cells residing in this intestinal lamina propria. So, when we isolated lamina propria cells from the intestinal lamina propria and analyzed for the expression of CD11B and CD11C, three major subsets can be observed. And in the first part, we'd like to talk about this CD103 dendritic subset. So, probiotics are known to be beneficial in our health. And so, Hisaku-san and Jeon-san in my laboratory analyzed the mechanisms for the action of probiotic strains of the intestinal bacteria, especially Lactobacillus casei and the Bifidobacterium breve, under tight collaboration with the Yakulto Central Institute. So, since the intestinal bacteria has been shown to influence the development of T cells in the intestine, we analyzed the effect of these probiotics on the T cell development in the intestine. So, for this purpose, B6 mice were orally treated with the B. breve or L. casei every day, and 3 months after the treatment, intestinal T cells were analyzed for the production of IL-10, IL-17, and interferon gamma. So actually, in the case of the L. casei treated animals, no remarkable change cannot be observed. But in the case of the B. breve treated mice, production of IL-10, IL-17, and interferon gamma from CD4 T cells was not altered in the small intestine. But when we see the large intestinal CD4 T cells, IL-10 producing cells, the number of IL-10 producing cells was tremendously increased in the large intestinal lamina propria of the B. breve treated animals. So actually the composition of fecal microbiota was not altered in B. breve treated mice. B. breve seems to directly affect on the IL-10 producing T cells in the large intestine. So IL-10 is mainly produced from the regulatory T cells in the intestine. So we next treated FOXP3 GFP animals with the B. breve every day. And 3 weeks after the treatment, IL-10 production was analyzed by intracellular staining. So in the case of the FOXP3 positive populations, B. breve treatment did not increase IL-10 production. But when we see the FOXP3 negative population, IL-10 producing T cells was tremendously increased. So IL-10 producing FOXP3 negative populations are known to be the T regulatory 1, so-called TR1 cells. And these TR1 cells are characterized by the expression of CMAP, IL-21, and aryl hydrocarbon receptors. And in vitro studies have indicated that these IL-10 producing TR1 cells can be induced by both IL-10 and IL-27. So we next analyze these IL-10 producing cells are the TR1 cells or not. So for this purpose, CD11C positive dendritic cells were isolated from the intestinal lamina propria, and these cells were first treated with B. breve. And after 24 hours of the treatment, these cells were co-cultured with spleen-derived naive CD4 T cells. And after 4 days of the co-culture, T cells were stimulated and analyzed for the expression of CMAF, IL-21, and AHR. So CD4 T cells co-cultured with L. casei treated CD11C positive dendritic cells. Expression of these TR1 specific markers was not increased at all. But in the case of the CD4 T cells, co-cultured with these P. breve treated dendritic cells, expression of CMAF, IL-21, and AHR was markedly increased. These findings indicate that B. breve induces IL-10 producing TR1 cells via activation of intestinal dendritic cells. So, intestinal dendritic cells can be divided into the 2 major subsets based on the expression level of the CD103 or CX3CR1. So we wondered which subset of dendritic cells are responsible for the B. breve dependent induction of TR1 cells. So we isolated CD103 positive dendritic cells or the CX3CR1 positive dendritic cells from the intestinal lamina propria and treated with B. breve and co-cultured with the naive CD4 T cells. So in the case of the CD4 T cells, co-cultured with the B-breve-treated CX3CR1 positive dendritic cells, IL-10 production was not increased at all. But in the case of the CD4 T cells, co-cultured with CD103 positive dendritic cells, B-breve treatment enhanced IL-10 production. So these findings indicate that intestinal CD103 positive dendritic cells are required, responsible for the B-breve dependent TR1 cell induction in the large intestine. So these TR1 cells have been shown to be induced by IL-10 and IL-27. So we next analyze whether these cytokines are involved in the B-breve dependent induction of TR1 cells in the large intestine. So first, we analyzed involvement of the IL-10 in the TR1 induction. For this purpose, we isolated intestinal CD103 positive dendritic cells from wild type or the IL-10 knockout mice and treated with B-breve, and then co-cultured with the naive wild type CD4 T cells. So in this case, CD4 T cells co-cultured with the B-breve treated IL-10 knockout dendritic cells do not become the TR1 cells at all, indicating that DC-derived IL-10 is required for the B-breve dependent TR1 cell development. So we next analyzed the involvement of the IL-27. In this case, we used naive CD4 T cells derived from IL-10 T receptor knockout mice. So we isolated CD103 positive dendritic cells from wild type mice treated with B-breve, and then co-cultured with naive CD4 T cells derived from the wild type or the IL-20 receptor knockout mice. So CD4 T cells from the IL-27 receptor knockout mice were defective in the induction of TR1 cells as shown here. From these findings, we can say that B-breve acts on intestinal CD103 positive dendritic cells to produce IL-10 and IL-27, and thereby induces IL-10 producing TR1 cells in the large intestine. So in the next experiment, we analyze whether these IL-10 producing TR1 cells are responsible for the maintenance of the gut homeostasis. So, immunocompromised SCID mice, when these mice were transferred with CD4 positive, CD45RB high T cells, these are the so-called naive T cells. So SCID mice transferred with the naive CD4 T cells suffered from this very severe intestinal inflammation, and they showed severe weight loss. But when these mice were orally treated with B-breve every day, B-breve treated animals did not develop severe intestinal inflammation, and they did not suffer from the severe weight loss. Thus, B-breve treatment ameliorate the intestinal, actually T-cell dependent intestinal inflammation. So we next analyze this amelioration of intestinal inflammation is dependent on the IL-10 production. So we first analyzed, we first isolated chronic tissues from the B-breve treated animals and cultured for 24 hours. And after that, supernatants were analyzed for the IL-10 production and inflammatory interferon gamma production. In B-breve treated animals, IL-10 production was highly enhanced, and inflammatory interferon gamma production was severely reduced. In the next experiments, we analyzed whether this IL-10 is actually derived from the T cells. So, for this purpose, immunocompromised SCID mice were then transferred with the naive CD4 T cells derived from IL-10 knockout mice, so actually defective in the IL-10 production. So in this case, B-breve treatment did not improve severity of intestinal inflammation at all, and they remain suffering from the severe weight loss. These findings indicate that T-cell derived IL-10 is required for the B-breve dependent amelioration of intestinal inflammation. So, in this study, we can show that B-breve acts on the intestinal CD103 positive dendritic cells to induce IL-10 producing TR1 cells, regulatory cell subset. And this TR1 cell subset is responsible for the suppression of intestinal inflammation. So, in addition to these CD103 positive dendritic cells, we have previously characterized this double positive population as the CD70 high CX3CR1 positive dendritic cells that mediate commensal bacteria dependent in the action of, in this case, Th17 cells in the large intestine. Thus, this double positive population includes CD70 high CX3CR1 positive dendritic cells inducing Th17 cells. But, as you can see here, this is a very large population when compared with other subset of the innate immune myeloid cells. So we wondered if there is other subset of the innate immune myeloid cells within this population. So we asked Hisaku-san to analyze this double positive population precisely. And what she found was that this double positive population can be further divided into the three subsets based on the expression level of the CX3CR1. So actually CX3CR1 negative, intermediate, and high population. So she decided to analyze the differential function of these three subsets. And she found that the negative population of myeloid cells are the dendritic cells inducing Th1 cells in the large intestine. And she found that this CX3CR1 intermediate population are the CD70 high dendritic cells inducing Th17 cells. So she further analyzed the function of this CX3CR1 high subset. Unlike other these subsets, this CX3CR1 high subset did not induce Th1 cells, Th17 cells, or regulatory T cells. No induction of helper T cells or regulatory T cells. But she found that this CX3CR1 high subset of myeloid cells suppress T cell proliferation. So, when CD4 T cells, spleen-derived CD4 T cells, were co-cultured with dendritic cells with a various number of the regulatory T cells or the CX3CR1 high myeloid cells, actually regulatory T cells suppress T cell proliferation in a dose-dependent manner, because these regulatory T cells are suppressing T cell responses. Similarly, this CX3CR1 high subset of myeloid cells also suppress T cell proliferation in a very similar manner that observed in the regulatory T cells. Thus, in vitro studies have indicated that this CX3CR1 high subset of myeloid cells suppress T cell proliferation. So our next interest is the in vivo function of this CX3CR1 high subset of innate immune myeloid cells. So, again, immunocompromised SCID mice were transferred with naive CD4 T cells. And these mice were suffering from the severe intestinal inflammation, and they showed severe weight loss. But when this CX3CR1 high subset of myeloid cells were transferred into these SCID mice just one day after the naive T cell transfer, this transfer of CX3CR1 high subset of myeloid cells ameliorate the severity of intestinal inflammation. And these mice did not suffer from the severe weight loss. Thus, transfer of the CX3CR1 high subset of myeloid cells suppress T cell dependent intestinal inflammation. So, in the next experiment, we analyzed T cell proliferation in these mice. So, naive CD4 T cells were isolated first, and then labeled with a CFSE, and transferred into, in this case, immunocompromised LAG2 knockout mice. And 7 days after the transfer, CD4 T cells in the colonic lamina propria was analyzed for the CFSE intensity. In LAG2 knockout mice transferred with naive CD4 T cells alone, CFSE intensity was greatly diminished indicating that transferred CD4 T cells robustly proliferated in the intestinal lamina propria in situ. In contrast, in LAG2 knockout mice, co-transferred with CX3CR1 high subset of the myeloid cells, CFSE intensity remained very high. These findings indicate that this transfer of the CX3CR1 high subset of myeloid cells suppressed proliferation of the transferred T cells in the large intestinal lamina propria. So, we next analyze the localization of this transferred CX3CR1 high myeloid cell subset. So, CX3CR1 high cells were isolated and labeled with a green dye and transferred into the mice. 2 days after the transfer, immunohistochemical analysis was performed. So green dye labeled cells could not be observed in several lymphoid tissues, including spleen, mesenteric lymph nodes, or the Peyer's patches. But these green dye labeled cells could be easily observed in the intestinal lamina propria. So actually, green dye labeled cells can be observed just beneath the epithelial cell layers of the intestinal lamina propria. From these findings, we can say that CX3CR1 high subset of myeloid cells, we would like to call this subset as regulatory myeloid M-leg cells. These M-leg cells suppress T cell proliferation in the intestinal lamina propria in situ and thereby inhibit T cell dependent intestinal inflammation. So, in the next experiment, what factors regulate this unique function of regulatory myeloid cells? We found that the number of the M-leg cells is normally observed in the IL-10 knockout mice, which develop intestinal inflammation. We isolated M-leg cells from the IL-10 knockout mice and analyzed for the suppressive activity for the T cell proliferation. Actually, wild-type M-leg cells suppress T cell proliferation, but in the case of the IL-10 knockout-derived M-leg cells, suppressive activity was totally gone. Since STAT3 is essential for the IL-10 signaling in the myeloid cells, we also analyzed STAT3 knockout-derived M-leg cells. Similar results can be obtained. In the case of the STAT3 knockout M-leg cells, suppressive activity was severely impaired, unlike the case in the wild-type M-leg cells. Thus, in vitro studies have indicated that in the absence of the IL-10 STAT3 pathway, suppressive activity of T cell proliferation was severely impaired. So my next interest is the in vivo function of these STAT3 knockout M-leg cells. So again, LAG2 knockout mice transferred with the live CD4 T cells suffered from the severe intestinal inflammation, and transferred of the wild-type M-leg cells ameliorate the severe intestinal inflammation. But in the case of the STAT3 knockout M-leg cells, these STAT3 knockout M-leg cells cannot prevent development of the intestinal inflammation. Thus, in the absence of the IL-10 STAT3 pathways, suppressive activity of the M-leg cells was severely impaired in vitro and in vivo. So, in the first part, we told that in the absence of the STAT3 or IL-10 signaling pathways, dendritic cells, innate immune myeloid cells such as dendritic cells were abnormally activated to induce aberrant Th1 or Th17 induction through the high induction of IL-12P40. And these aberrant Th1 or Th17 responses is responsible for the development of inflammatory bowel diseases. But in this study, we found that in the absence of the IL-10 STAT3 pathways, suppressive activity of newly identified these M-leg cells was also impaired. So we wondered if this impaired suppressive activity of M-leg cells was involved in the pathogenesis of the inflammatory bowel diseases in the STAT3 mutant mice. So, for this purpose, to analyze the involvement of the M-leg cells in the pathogenesis of the inflammatory bowel diseases, STAT3 mutant mice, actually recently STAT3 fox mice, were transferred with the wild type normal M-leg cells 2 times. And 2 weeks after the last transfer, we first analyzed Th1 or Th17 responses by determining the production of interferon gamma or the IL-17 from the splenic CD4 T cells. So actually, in the STAT3 mutant mice, Th1 and Th17 responses was highly enhanced. Even in STAT3 mutant mice, transferred with naive normal M-reg cells, Th1 and Th17 responses remained very high. Under these conditions, we analyzed the development of the inflammatory bowel diseases. So actually, STAT3 mutant mice developed severe intestinal inflammation. But STAT3 mutant mice transferred with normal M-reg cells did not develop severe intestinal inflammation. Thus, defective activity of M-reg cells is actually involved in the pathogenesis of the inflammatory bowel diseases when T cells were overactivated. And even when T cells were overactivated, transfer of the normal M-reg cells can prevent development of inflammatory bowel diseases. Thus, intestinal regulatory myeloid cells, M-reg cells, show the IL-10 STAT3 dependent suppression of T cell proliferation and thereby inhibit intestinal inflammation. So, as I told you, there are several unique subsets of the intestinal innate immune myeloid cells in the large intestinal lamina propria that mediate induction of inflammation or the suppression of inflammation. The activity of these innate immune myeloid cells in the intestine that interact with the microbiota is actually responsible for the maintenance of the gut homeostasis. Actually, in the absence of the microbiota, Th17 inducing activity of these subset are severely impaired. In addition, in the absence of microbiota, suppressive activity of these M-reg cells was also severely impaired. So by understanding how these innate immune cells activity can be maintained by the microbiota, we'd like to reveal how gut homeostasis were maintained by these innate immune cell subset in the future. So finally, I'd like to acknowledge the contributors in this presentation. So actually, the B-Breve project was performed under tight collaboration with the Yakult Central Institute, especially Professor Nomoto, and also performed by the Professor Kiyono at the University of Tokyo. And the M-reg cell project was performed under collaboration with the Professor Ishii and Professor Murakami at the Osaka University. So, in the very final slide, I'd like to make an announcement on behalf of the Japanese Society for Immunology. So, Japanese Society for Immunology holds the annual meeting annually, and the next annual meeting will be held in the next December, actually 1 year later. And under the leadership of the world-famous mucosal immunologist, Professor Hiroshi Kiyono at the University of Tokyo. And next annual meeting will be held all in English by inviting many foreign speakers from the United States, Europe, and Asian countries. So actually, in the next annual meeting, we do not use Japanese, only English, to facilitate, for the creation of new international relationship worldwide. So, on behalf of the Japanese Society for Immunology, I'd like to ask all of you to submit an abstract. So actually, we are waiting for your participation. So thank you very much for your attention.